Good morning, everyone. Thank you very much for joining today's webinar, the webinar that Omixon is presenting about epitope matching. There is no excuse. This webinar is a collaboration between Omixon and Perch. My name is Effie Melista. I'm the head of product for Omixon, and my colleague Matthias Niemann from Perch will also be joining us as part of this presentation. We hope everybody is very well, and we can get started. Okay, so I will begin with a little bit of an agenda for what we are expecting for today. So we will be beginning with an introduction, then Matthias will be talking about the epitope matching approaches, then epitope matching misconceptions, a summary to close off with, and then we will be having some closing remarks and a question and answer session. So let's begin with a little bit of an introduction. First of all, some logistical things. Please stay muted. You will be able to see the little microphone on the left side of your name. Please keep that red in order to avoid any background noise during this presentation. If you have any questions during the presentation, please feel free to enter them into the questions box into your application, and we will make sure to answer all of them at the very end of the session. And finally, a recording of this webinar will be provided via an email. In that email, there is also going to be a short survey. And because our webinars are also ABHI accredited, you will be able to receive CEC credits if you submit the survey. So with that said, we can begin. So, Matthias?
Yep. Thank you, Effie, and thanks for Omixon for having me today. So you already mentioned the title of my talk. Yeah, so the idea is of today's talk to clear out some misconceptions about epitope matching and give you a brief overview on the different techniques. But first of all, let's take a step back, if you want so, to the cradle of the first HLA antigens. So when the first HLA antigens were discovered, basically what Jean Dosset, Jan van Root and Rose Payne saw was that antibodies reacted with some individuals, but not all of them. So basically you saw there's some sort of pattern of the reaction of antibodies. And from there on, and this discovery was actually a great deal, it was very clear that this is very important for tissue transplantation and organ transplantation. So why do I want to show this wheel here? Well, basically we iterated through that quite a couple of times. So we have our expectation and see something different in reality. We adjust our diagnostics and improve them, and then we have a new understanding of HLA matching. And after a couple of decades now, we are at a point where we know, okay, we need to do something more with a biological reasoning and with a biological structure in mind when it comes to HLA matching, and basically the name of that is epitope matching. There are quite a few different approaches for epitope matching, and therefore I want to go more into detail how the different pathways of allorecognition actually look like. And let's start with the Tcell recognition. Well, you have the pathway of direct recognition, where you have your reactive immune system, and this is here an example for solid organ transplantation, where you have foreign tissue and a working patient immune system. And basically the T cells could detect the foreign HLA on the donor antigen presenting cells. And if you consider the indirect pathway, this looks a bit different. You do not directly, or the T cells do not directly detect the foreign HLA, but you have processed donor material, so short fragments, peptides and stuff like that, that are endocytosed and presented on patient HLA to the patient T cells. So there's no direct detection of the antigen, but well, therefore it's called indirect. Then there's this funny thing that's somewhere in between that's called semi-direct T-cell allorecognition. And the idea here is that on the right-hand side you still have basically an indirect pathway, but on the left-hand side you see there's a direct portion where the foreign HLA of the donor is transferred onto the patient APC via exosome fusing or just via mechanical contact, and then the HLA is stripped off and presented on the patient APC. And the interesting thing about that is that due to the close proximity of the T cells now and the HLAs that are activating the T cells, the T cell help of the CD4 T cell help to the CD8 T cell is much more pronounced than because these two cells are so close to each other. That's why I wanted to mention that here. If we look on the B-cell side, we have a T-cell independent B-cell activation. So basically the B-cell receptor gets activated by some antigen, and in consequence it gets activated and it's doing its job basically. But we also have a T-cell dependent pathway, and that's pretty interesting as it is a two-step approach where the B-cell receptor, so the purple guy here, is detecting basically the foreign HLA, so patches of the HLA actually, and via this activation of the B-cell receptor, an endocytosis process starts where the foreign HLA is internalized, and then it's being processed in the patient B-cell, and eventually these peptides, foreign peptides, get loaded onto the patient's HLA, moved to the cell surface and presented to the patient immune system, where the patient CD4 T cell can then get activated and help the B cell, or telling the B cell that this was actually an interesting antigen that the B cell should react against. So it proliferates and is doing its job. So basically this is something we want to avoid in transplantation medicine. So let's now look how these epitopes look like, and also in my talk I will use these 3D charts here, or these plots. So B-cell epitope is basically characterized by being a patch or patches, different patches on intact foreign HLA. Not necessarily foreign, so your own HLA also have applets, of course, but these patches, these surface areas are considered as being an epitope, or well, today we call it applet usually. And what we see here is an HLA A021 with a 145 HKA, so that's basically the name of that red applet we see here. And they do not necessarily need to be linear or continuous. As you can see here, these positions 144, 145 and 149 are not necessarily one by one next to each other, and they are rather short or small actually. When it comes to surface accessibility, well, it's not entirely understood. So this example, what I showed you here, is actually very well accessible, of course, but you also find applets close to the cell membrane, and it's not entirely understood why these are, or if we could consider these surface accessible, but still they are very dominant when it comes to being reactive. So how does a T-cell epitope or an indirect T-cell epitope now look like? Well, that's a different story. So we do not consider the actual intact HLA here and its peptides, but the fragments of digested HLA. So that means the actual Tcell or indirect Tcell epitope, you won't find it on the, or necessarily on the surface of the foreign HLA, but it will be presented in the binding groove of your own HLA, or on the reactive immune system's HLA actually. And these peptides are linear N-mers, and depending on HLA class, this could be either something around 9 mers if you consider class 1 with a rather closed binding groove, or around 15 mers if you talk of class 2 presentation, where the binding groove is a bit more open, and therefore it could also be sometimes a 16 or 17 mer or something like that, so it can stretch outside of the binding groove in the end. So with that said, let's look what the different models are actually, or how they are named and what's the difference. So let's start with the Bcell epitopes and the Bcell recognition. So basically it all started with these Terasaki epitopes, where the 1 lambda team basically, or Terasaki himself, tried to identify certain reaction patterns of the antigen or single antigen tests, or antibody tests actually, and understand why there are certain cross reactions and stuff like that. Based on that, or well, going into the same direction, René Duquesnoy and the HLA matchmaker defined the applets more in a systematic way, and defining the same amino acid patches on different HLA molecules. The EMS 3D model, which is of the Cambridge group around Kosmoliapsis, now gives a bit more characteristic to these applets or epitopes by defining some physiochemical properties. So we again have a new definition of what is exactly now a mismatch and what not. So only because the amino acids mismatch, it's not necessarily a bad mismatch. And pretty recently the Leiden group published the HLA EMMA tool, which is taking this approach further, the Bcell recognition, by using some more bioinformatics tools on solvent probabilities, or surface accessibility in solvents. So this is also quite interesting. On the direct T-cell recognition side, we have the well-known DP T-cell epitope model from Katharina Fleischhauer, which is well known and applied in stem cell transplantation if you are beyond 10 out of 10 matched unrelated donor selection, and the histocheck model from the Hanover group that tried to define a couple of surface amino acids and their mismatch or amino acid mismatch score basically. As you can see, they are somewhat overlapping also with the indirect pathway, because you also and always need to keep in mind that we are looking on the same things here, on the same amino acid sequences. So there is obviously some overlap between the different techniques because we're looking on the same data. And ultimately, for the indirect T-cell recognition, there's now the Perch model that tries to model that. And let's check out what that all means clinically. Well, you all know applets in kidney transplantation are a thing. They are correlated to transplantation outcomes, DSA and graft survival. I just picked 2 transplantations here, publications here, because they were just lying on my desktop actually, but there are dozens out there or even more. And what I want to emphasize here is especially that there is evidence for this effect being statistically independent. So this is very important to keep in mind, that we are beyond classic HLA matching, actually. However, as you see, or as I mentioned before on the slide, there are a couple of different models. So you have different definitions depending on publication. Some use antibody verified applets, and some use all the applets. So it's not entirely clear yet what to do with that. When it comes to Perch, we also see a strong correlation with de novo DSA development and with graft survival. And as said for the applets as well, we also see for the Perch score that you have an impact, a large impact beyond HLA matching. So like we see here in the publication from Niels Lachmann here in Berlin, so even if you have a single mismatch on the DR locus, for example, you can well discriminate with a Perch score between a lower risk and a high risk for developing DSA against that particular mismatch. What also is very important, I think, is the fact, and that's shown here by the group of Eric Spearings and Kirsten Genökelik in Utrecht, that this is an effect not only short-term after transplantation, not only hyperacute or acute rejection, but it's also important for the chronic rejection. So even after a couple of years, you see increased hazards by selecting a donor with a high Perch score. So obviously, and we get that question quite often, what should we do now? Should we use applets or should we use Perch or both, or what to do? What's better? And this is, well, let's say it's complicated. Even in the same populations, we see different, or we see a good correlation between the matchmaker and DSA and Perch and DSA, and depending on the locus, depending on the setting, it varies a little. But very important to keep in mind is that, that's also the table down here of the publication of Niels Lachmann, is that in a multivariate analysis, you see that both the matchmaker and the Perch score survive the multivariate analysis. So that means both have their unique and individual impact on the predictive capability of epitope matching. And just to be consistent here, classic HLA matching is already far beyond then, and it's already covered by both matchmaker and Perch together. So you have to keep in mind, and that's why I also wanted to show you the different pathways in the beginning. It's not just an alternative way of epitope matching to use now indirect Tcell epitope matching and Bcell epitope matching, but it's important to keep in mind that CD4 Tcell help is mandatory for naive and memory DSA responses, and that chronic allograft rejection is largely mediated by the indirect pathway. And those aren't quotes of myself, but of these papers here. So it is well known that these different concepts work well together and are somewhat dependent on each other. So the ultimate answer will probably not be applets or Perch, but applets and Perch. And just to give you a glimpse, idea what we are working on and how it will, where we will go to in the future. Here an abstract of 2017 it was, I think, at the FE meeting, where you can see, or where we tried out a new prototype where you apply a subsequent Bcell epitope or HLA matchmaker matching and a Perch matching. And as you can see, the predictive performance of this model is then beyond the Perch model and beyond the matchmaker model. So this indicates we are moving towards an and not an exclusive or. So let me jump now to stem cell transplantation to be complete here. For stem cell transplantation, we are with Perch looking into the graft versus host directed Tcell allorecognition, and the model then looks a bit different, as you can imagine, because we now have a new immune system transplanted into our patients. So the direct recognition now would be that our donor T cells react against the patient APCs or the HLA on the patient APCs, and the indirect pathway is a bit more interesting here. because these internalized peptides of mismatched patient HLA is especially interesting if it is presented on HLA that is matched or shared between the donor and the patient, because as you can imagine, you still have plenty of patient APCs floating around that speak basically the same language as the donor T cells if the HLA between donor and patient is matched. And therefore, you always need to consider especially these HLAs that both have in common. So how does that look in the clinics? Well, in 9 out of 10 matched unrelated donor transplantation, you also see here a correlation between the Perch score and graft versus host disease, and this then strikes through on overall survival and on relapse mortality. And what's maybe also very important here is to keep in mind this paper from 2014 already, where the group from Utrecht showed that even beyond TCE compatibility, it is still a good idea to go for a low Perch score because it still could discriminate cases with lower incidence of GVHD compared to the ones with increased risk and a higher Perch score. And this, it explains again, or you can explain that effect by going back to the models again, and as both concepts try to describe different pathways of allorecognition, it makes perfect sense they have their unique value. So the next question that usually comes then up is, but what about Haplo? So with Haplo, it's a bit more complicated and a work in progress situation, I guess. So there is a publication from the US from last year where we see a correlation of the Perch score with GVHD after a Haplo stem cell transplantation with a post-transplant cyclophosphamide protocol. However, before that, a Chinese group published that this does not work in an ATG-based protocol. So probably the treatment has here an important effect, and we are not entirely sure to what extent this impact actually has to be taken into account. And what's also very important is, and that's where I mentioned this shared concept in particular, is that for haplo transplantation, you have way less shared HLAs, so you have less cells that talk the same language as those donor T cells. So there's a counterintuitive thingy, meaning that more matches means more presenters, and therefore probably a higher Perch score than if you wouldn't have any matching alleles at all. So rather interesting. All right, so let's come quickly to the second part here, the misconceptions, actually. So there are a couple of things that we always hear at conferences, and I quickly want to dive into these. Point one is epitope matching needs high resolution typings. Point two is epitope matching is too complicated to do. I don't have time for that. And epitope matching isn't considered in donor selection, so why should I care? And the fourth one is, well, it's just not ready yet. So let's dive into these and let me give you some answers on that. First one, well, not necessarily right. So there are approaches of multiple imputation that's also available in the Perch platform, where you can use serologic split or molecular low typings to extrapolate the most probable high resolution typings, and then you match all of them, all of the different donors and patients side by side, and aggregate the result in the end. And we see in a paper from 2017 that it does indeed make sense, and the deviations which you get are clinically, for 99% of the cases, not important. So, well, actually, you could use low-res typings. It's too complicated. Yeah, well, you could argue on that. Actually, it has never been easier, to be honest. The Bcell epitopes are available in 1 lambda fusion, Immucore match it for antibody analysis to explain the reaction patterns. The DP-TCE models available as a web service, and also the Perch service is available online and integrates now with all the major vendors' software. So just to show you here how it looks like in HLA Twin, you have that nice Send to Perch button up there, and then you can submit your typing directly to the Perch website. Then you jump there and you have all your typing data then on the website and can do whatever you want there in terms of epitope matching. And if you aren't using these tools, you can still use our semi-automated data entry stuff like pasting GL strings using CSV, HML, so all these common standards. So these data entry stuff, I think it's not a valid argument anymore. And also you can see these nice plots, so aggregated data. So you do not necessarily have to be a huge expert on that to still get a good idea about the epitope match grade. So that was point 2. Point 3, epitope matching isn't considered in donor selection, and that's especially the case for postmortem kidney allocation. But however, I want to stress that we are working on that, and there are a couple of groups working on that, and we did some simulations on the Eurotransplant kidney allocation system. This is here a work in progress of a couple of abstracts that I presented already, where you can see that if you apply Perch in the ATCAS allocation system, you can squeeze the high-risk patients down to a lower risk group in terms of epitopes, and this holds for all the HLA match grades. So basically it's a good idea to do that, but this is of course still a work in progress and has to be included, that's right. However, you could also use something what we call the risk and acceptable mismatch profile, where you match 10,000 virtual donors with a patient to get a good idea about the donor frequency in terms of epitope mismatch probability. And if you aggregate this with the single antigen beat MFIs, this is a great tool for defining unacceptables. Like you see here on the screen, on these plots, you have the Perch scores in the blue bars and the MFIs in pink, and now you can make a good selection of what the patient shouldn't get in terms of HLA mismatches. So I guess the future is some epitope-supported antibody diagnostics, and this here again shows that it's important to consider all of the different models because your antibody diagnostics, so actually finding those antibodies, is only one thing, one step. It's a snapshot of the current situation, and you need the matchmaker to understand these patterns and allow extrapolation for other antigens that are not part of your test. And then you can explain a bit these reactions and make a good decision for unacceptable mismatches. However, Perch in that case would then be a very useful tool to add a risk factor to all these alleles that are yet antibody negative, because you want to transplant the antibody negative donors. Still, if they have a high Perch score, they again have a high risk of the patient developing antibodies pretty soon. So you need to keep that in mind. And basically, with his custom population feature, which is out there since last year, you can pretty good focus these analysis on your own custom local population and not just some generic one. So this is also clear. And the fourth one, it's not ready yet. Yeah, well, that's actually a good point to close the circle to my introduction of the history of HLA. It is, of course, an iterative process, and these algorithms are still in improvement. However, we actually know that epitopes are a thing because they're also part of the upcoming IHLW. However, we know there is scientific evidence, and we need to start pretty soon with it to understand more how that's all working and already using this improved predictive capabilities, I guess. So that's basically it. I want to stress out again, even if we cannot answer your questions now live, or if you want to dive more into detail, just visit the Perch or Omixon website. here's our Perch Education tab. There are a couple of webinars and recordings, and also this literature compass to dive into or see which publications are out there on the different domains. And yeah, Effie, I guess you also want to tell a couple of things.
Thank you, Matthias. Yes, absolutely. Thank you very much for the very detailed and educational session here. So just like Perch, we also have, if you are looking for more information for NGS genotyping and not only your software, please feel free to use the Omixon website. There is also a large number of publications, past webinars, and other material that you could potentially utilize. And then thank you very much for attending. We are ready to actually get questions now. So feel free to put in your questions in the questions box. And we definitely have a few minutes for that. Okay, so one of the questions that we have here says, one of the issues that was not mentioned is that the list of eplets vary from one group to the next. Some are not proven. How do we deal with this?
Yeah, this is a good point. I mentioned that earlier as well, and it's complicated to answer that. Sooner or later, you have to, of course, decide for the one or other model. Right now, it looks like these antibody-verified applets are one way to go. On the other hand, yeah, I guess we just need to decide for one and then apply that and learn from there on. I guess it's not a good idea to just wait now until we finally know what applets we need to use, because it is a learning process, and just by using, we will learn more, I guess.
Yeah, and there is also one more follow-up on this. And how do we apply, or is it possible to apply this in the context of platelet transfusion?
Yeah, that's a good one. So there are plenty of, or I receive this question quite often, and we are very interested in that. The thing is with platelet transfusion, I guess you need plenty of data, and I didn't see any publication out there that's on platelet transfusion. So I would be very interested to collaborate on that end, to be honest. So if someone's out there with a good data set on platelet transfusion, I guess it makes sense to take a look, because biologically this should make sense, but we didn't see that clinically yet.
Mm-hmm. Okay, next question. How do you deal with the different versions of applet database?
Yeah, that relates probably to the question before of the different definitions of applets. I guess ultimately it will be something like the IMGT database that you need to have some versioning, and then your applet results are only valid for a particular database. And if that database changes, your definition of applet mismatch is then also changing. But I mean, that's basically the same with any in vitro test. That's also only working in its own scope. And with a new version of that in vitro test, for example, the single antigen beat assays, you may also get different results then. So I guess it's fine.
Okay, so I think we have actually gone through a few questions. Thank you very, very much, everybody, for joining us today. If you have any further questions, by all means, feel free to join, to email either Omixon or Perch directly, and get in contact with us anytime. We will be happy to answer any questions or initiate any potential collaborations on some of the paths that have not been quite studied yet, as Matthias said. So Thank you very much. We are looking forward to your surveys in case you would like to get CC credits for this session. And we are looking forward to seeing you again next time. Thank you and have a great day.
Bye.